Okay, let's move on to the second part of the lecture. So here's a thought question. If capillaries are all sort of improvised, even in the normal tissues of our body, then it must be that there are limits to the growth of our capillaries, because there must be some way that our normal tissues balance how extensive the capillaries should be through them so that they're nicely, properly spaced out in our tissues. And at a certain point, you want to prevent additional capillaries from keep growing, right? Otherwise, they would become like that messy capillary network in the tumor. So what limits angiogenic signals from normal cells, right? So what's keeping normal tissues from always signaling for more capillaries to grow? And how do normal healing tissues, after a wound is being healed, how does the wound or the healed tissue know when to stop stimulating new capillaries to grow? So there are three main inhibitory mechanisms that we're going to talk about that limit vascularization and limit angiogenesis. And these have to do with the presence of enough oxygen reaching tissues, because obviously if enough oxygen is reaching tissues, then that could be used as a sign that you don't need to have more capillaries growing. Um, there are certain molecules that are released by endothelial cells, such as TSP1, that are inhibitory, that actually inhibit the capillaries from growing anymore. And also just the availability of things like VEGF and mitogenic signals is a way of controlling how many of these stimulatory signals actually are allowed to be received by things like endothelial cells. So let's start off with the first one. We've already talked about this before, so you've seen this. This goes back to chapter 7 when we were learning about different types of tumor suppressor genes. So we learned that we have a protein known as HIF1 that helps regulate things like the expression of the VEGF protein. So HIF1 normally is acted upon by an enzyme, which is a hydroxylase, that if you have normal levels of oxygen surrounding these cells, then this hydroxylase will modify the HIF1 protein by adding these hydroxyl groups, and then this modified HIF1 then becomes a target of this VHL tumor suppressor gene. This VHL then labels the HIF1 by ubiquitination, and that leads to the degradation of the HIF1. So again, just like when we talked about beta-catenin, this is another example of the way our cells are engineered. They constantly produce this HIF1 because they always want to be ready to turn on things like angiogenic signals if suddenly they need it. But normally, if there's enough oxygen around, they just simply degrade the HIF1 so that you don't have angiogenesis. But in low oxygen conditions, hypoxia, then the HIF1 is allowed to function and activate, because you won't have it modified by the proline hydroxylase because the levels of oxygen are too low. And as a result, you'll have more HIF1 that activates. It's a transcription factor that binds to the regulatory regions of genes like VEGF. So in this particular case, just the amount of oxygen that's present can regulate whether VEGF gets turned on to stimulate additional capillary growth. One of the side effects of a tumor getting very large, as I mentioned before, is that it puts a lot of physical strain and stress on the surrounding stroma. Here we have a nice illustration of a tumor cell down here. In this particular case, you can see the tumor cells are very densely packed in there. This dotted line is the boundary line of where sort of the basement membrane is still present. And then outside of it would be the surrounding tissue of the stroma. And you can sense again that the surrounding tissue is being heavily squashed. Okay. A lot of that has to do with tumors because of the leakiness of all the capillaries in them. They have very high hydrostatic pressure. In other words, the water pressure inside the tumor is greater than the water pressure in surrounding tissues. And that means that there is a lot of pressure being exerted on the surrounding stroma. This can cause things like the surrounding lymph vessels which are indicated by the arrows, to collapse and actually be kind of shredded or torn apart, meaning that a lot of fluid and material out here is being drained now by these lymph vessels that no longer just have very carefully designed little openings in them, right? They're all kind of torn open, so a lot of things can drain in them. This can also result in leaky capillaries. And a lot of PDGF signal being released by all these tissues being kind of squashed and torn apart. And so that actually creates a situation where you have lots of tissue building up out here. Right? So this high hydrostatic pressure then means that there's a tendency for water to be squeezing out of the tumor. So here's a question. How might this impact drug treatment of this particular tumor? In other words, if you imagine that this tumor is in the body somewhere, right, and the patient is being given some sort of drug, a therapeutic drug to treat this tumor. Let's say the person takes it either orally or even IV through their arm. Then this medication has to flow through the blood, and you're hoping that some of it's going to get to this tumor. Right? But if the medication arrives, how might the fact that the tumor has much higher hydrostatic pressure and the water is constantly being shoved out of the tumor, how do you think that might impact the ability of the drug to penetrate the tumor? Does anybody have any thoughts on that?
Less drug would be able to go into the...
Yeah, exactly, right? So you would actually have less drug actually getting into the tumor. And so that means you'd need to have a much higher dose of drug given to the patient just so that the tumor actually gets targeted by sufficient dosage. So that causes all kinds of problems because meanwhile, the other normal tissues in the body are exposed to much higher levels of that drug, and meaning that the patient would be subjected to higher levels of off-target side effects, potentially cytotoxic side effects of the drug. So that's a real problem then with a lot of systemically given drugs that are meant to be small chemicals or compounds that only work if they can actually get into the tumors. All right, so another way that cells or tissues regulate capillary growth is by actually using inhibitory molecules such as this TSP1. So TSP1 is a signal that signals apoptosis to happen. When you have a section of capillary, let's say we have a normal section of capillary here, so these are older endothelial cells. This capillary has been around for a little while now. They're surrounded by these pericytes that helps structurally support the capillary walls, prevent it from being too leaky, for example. If you look at one of these mature endothelial cells, this mature endothelial cell is just doing its job as being part of the wall of the capillary. Somewhere at the very end of the capillary, you might find you have newer endothelial cells that are growing. So a recently formed endothelial cell, like you might find closer to the tip of the end of a growing capillary that's maybe growing towards a wound in response to VEGF or something like that, they will express certain receptors. So one receptor is CD36, and CD36 responds or binds to this TSP1. So this TSP1 is being released by the mature endothelial cells that are up here, and only the newly formed endothelial cells have the receptor to respond to it. These newly formed cells will release FASL. FASL is a ligand for the death receptor. So these death receptors are present on the newly formed endothelial cells, and then the newly formed endothelial cells then actually receive this FASL signal, and as a result are triggered to undergo apoptosis. So that means if you have a lot of mature endothelial cells present already, that implies that your capillary network or the capillary bed probably is in pretty good shape, and so you're actually inhibiting new endothelial cells from forming and causing the capillary to branch out or grow. Whereas the mature endothelial cells are not responsive to the fast cell that they are stimulated to produce. A third mechanism is simply the fact that if you have an intact basement membrane and intact healthy stroma, then many of the mitogenic signals that are being released by cells to try to stimulate angiogenesis end up just being caught or held in the extracellular matrix. So that includes signals like VEGF. So here's a really nice image. This is one of my favorite images from the book, because here you have a tumor cell that, or a tumor, a small tumor. Maybe it's just a few hundred, maybe 1,000 cells at this point that's growing. So those are the cells that are in dark blue. And this tumor has now reached the point where it's large enough that the cells in the middle are getting stressed out, right? Because they're getting too far away from the surrounding capillaries. So maybe it used to be that this brown thing on the right over here was the original capillary that was kind of flowing by here. So maybe here's another capillary, here's another one, here's another one. These tumor cells then have reached a point where they are beginning to undergo hypoxia in here. And as a result, they've managed now to kind of stumble upon different mutations that are releasing things like VEGF. So that VEGF then, right, is managing to get out of here somewhat through the basement membrane. So you can really see how this tumor is still contained by the basement membrane. Right? And so some of that VEGF has managed to stimulate new angiogenesis. So new endothelial cells are now being stimulated to branch off of this nearby capillary and actually grow towards the tumor. But the important thing here is you see that the basement membrane is limiting the ability for the new capillary cells to actually penetrate and get to the cells in the middle of this tumor that really are starved of oxygen. So I think this is a wonderful illustration, because at this point then, the tumor is stuck. It really can't get much bigger because of its basement membrane sort of holding on to a lot of the VEGF signals and other mitogenic signals, and also the basement membrane preventing physically the new capillaries to grow in. So this suggests that the next step for this tumor to be able to really suddenly grow and expand has to do with breaking free of these limitations of the basement membrane, right? And importantly, being able to then really stimulate the growth of capillaries to supply a new capillary network inside of this tumor mass. And so we call this the angiogenic switch. And so it's very much a turning point where a tumor has gotten to a size where it's limited by its size. It really can't grow much further unless it then stumbles upon those mutations that allow it to do this angiogenic switch, which involves the breakdown of the basement membrane and, importantly, the sudden attraction or stimulus of capillaries to be able to penetrate and grow into the tumor. So you can actually think about then that those three ways of inhibiting angiogenesis that we talked about this lecture can be thought of as another way that we can apply the brakes on a growing tumor, right? That we can really inhibit a tumor from growing because we're limiting the ability of capillaries to grow. So tumors must break through the basement membrane in order to really manipulate the stroma further to encourage new capillary growth and actually perform this whole angiogenic switch, which is so crucial. So we can think about this in terms of this cartoon. This is a nice set of cartoons that highlight this. So here we have the original hyperplastic growth. So maybe this is in the pancreas. So you have a particular pancreatic islet group of cells in the pancreas that are now transforming. At first, they are hyperplastic. So we don't consider them tumorous. We don't consider them cancerous yet. They are forming a benign tumor. And this tumor might remain benign for years, potentially indefinitely, until they've stumbled upon this angiogenic switch. Right? So there are certain signals being sent out by these tumor cells as they learn more and more different sort of tricks through different mutations. And these are recruiting things like the precursor endothelial cells from the bone marrow, as well as other immune cells that are in the circulation. So you'll have mast cells and macrophages that then penetrate the stroma around the tumor. And you remember these macrophages like TAMs, tumor-associated macrophages, might get into the stroma and might be attracted to the wall of the tumor because of that wound healing response. Right? So at the top of the slide, I say, remember, there are parallels with tissue remodeling that take place during wound healing. So last time we talked about that these macrophages will remodel the stroma as part of wound healing, because as far as the macrophages think, oh, this is damaged tissue, it's a wound that needs to be cleaned up. So the old damaged tissue needs to be removed. So one of the things that the macrophages do is they release these MMP9 enzymes, these matrix metalloproteinases, that will digest away the fibrous protein network of the stroma, including the basement membrane. So here we have in gray, this is a VEGF molecule that's been sort of stuck in the extracellular matrix, like the basement membrane, because of these collagen, these pink collagen and lamin fibers. But now the macrophages digest those fibers away, the VEGF molecule is liberated, and that's a big critical part of the angiogenic switch, right? The basement membrane dissolves, these VEGF signals suddenly go up in level enormously to attract surrounding capillaries to start growing. And now these transformed cells, right, have successfully undergone the angiogenic switch, because now they have the opportunity to physically move out from the limitations of the basement membrane, and they're being supplied with the oxygen that they need because of all the capillaries that are now growing. So these nice images really illustrate that. So tumor invasion will greatly increase the vascularization in the area. And it goes back and forth, right? So the more vascularization you have, the faster the tumor will grow. So that's why you can really think of this angiogenic switch as a turning point, when suddenly the tumor went from growing pretty slowly, maybe over years, to suddenly now really exploding because of this positive feedback due to angiogenesis. So here is a early prostate cancer, prostatic intraepithelial neoplasia, or PIN. And remember, it's in situ, meaning it's still doing all of its dysfunctional mutations and things like that within the confines of the basement membrane. Right? So in the last lecture, we had that kind of funny analogy of these being the rebelling teenagers that are getting all crazy and rowdy in here, and they're no longer listening properly to the instructions of their parents, which are the normal stroma. But part of them getting all crazy is now they're sort of wearing their parents down, and the parents are getting confused and manipulated. So the stroma is becoming manipulated. And eventually then that really chaotic set of signals and the stumbling of the tumor cells upon those angiogenic switch mutations then suddenly allow massive vascularization to happen in the surrounding tissue. The basement membrane has broken down. So all of these tumor cells you now see have infiltrated the surrounding tissue. And there's no doubt about it, when you look at these dark brown stained capillaries, the network of capillaries has become far less coordinated and well managed compared to originally. Here below is human breast cancer, same type of example. When the breast cancer has gotten to a point where it's still in situ, you can really sense where the basement membrane is surrounding this, right? And these little dark dots are various capillaries, or maybe these are red blood cells actually indicating where the capillaries are surrounding it. Right? But once these tumor cells have turned on the angiogenic switch, then the basement membrane breaks down and you see far more extensive capillaries, allowing then these what used to be in situ tumor cells to now become full-blown cancer, right? And that really is the hallmark of when we switch from something being called a benign tumor or pre-malignant tumor to really becoming cancer, right? Once those cells have now found out and gain the ability to spread into the surrounding stroma.